Good evening, ladies and gentlemen. Thanks for joining. It's my privilege and great pleasure to welcome you today in 5th International Virtual Workshop on Research Methods in Public Health. As you're aware that this workshop will cover quantitative and qualitative research methods, appropriate investigation of problems in public health. Please join me in welcoming speaker of today's session, Dr. Harmanpreet Singh Kapoor. Dr. Kapoor is MSc Gold Medalist and PhD Statistics. He is working as Assistant Professor in Department of Mathematics and Statistics, Central University of Punjab. Dr. Harmanpreet Singh has got teaching experience of more than 5 years, and areas of his interest are actuarial statistics, risk models, financial mathematics, data science, and inferential statistics. I would like to mention here that he has coordinated and prepared 25 modules for the paper Statistical Application in Environmental Sciences for EPT Pathshala program funded by Ministry of Human Resource and Development and coordinated 4 field projects of Hindustan Mittal Energy Limited. He has got 6 research papers published, delivered 16 invited talks, and presented 2 research papers. Moreover, there are 17 MSc dissertations to his credit. It's our honor to have you, Dr. Harmanpreet Singh, as a speaker. So, without any further delay, I would request you to start your talk. Thank you.
Thank you, Dr. Bharti Kapoor. So I would like to thanks all the organizer for giving me this opportunity to share my views regarding this biostatistics, because basically I'm from statistics field and I'm also working nowadays in biostatistics. So I would like to share my experience with you. So let me share with you my presentation, then we will discuss about the topics. So I think my screen is visible to all of you, right?
Sir, and please make it full size.
It is already in full size, sir. Can you see this?
Yes, now it is fine.
Okay, fine. Okay, thank you. So most of you are I think I'm not aware whether all of you are aware about the term statistics or not, but I would like to give brief introduction about the term statistics, and I think all of you are already using the term statistics in general life, right? In day-to-day activities, like whether they have any data set and they want to get inference, or whether they want to you can say summarize the information, summarize the data set, and they by using a single value. So let us suppose I'm just would like to give brief introduction about the statistics. So in statistics, basically we generally statistics term is used that we have different tools and techniques with us that helps to extract information or draw inference from the data set, right? So we generally divide our statistics into two branches. One is called descriptive. And second is called inferential. Let me just- And second is inferential, right? In descriptive statistics, our main objective is that we have some data available to us, and we just want to represent that data set, right? So we have various tools like you can use measure of center tendency, measure of dispersion. So that things are not going to be relevant for our today's session. So main thing that I want to discuss with you that when we have a limited data set and we want to and where data set is coming from a particular population, that means we have a sample with us, and based upon the sample observation we want to draw inference regarding the population parameters. So in that case we use inferential statistics. So in inferential statistics we have two main objectives. One is called estimation, or you can say we have two further branches, and second is called testing of hypothesis. Okay. So when we have a sample with us, and our main objective is to find out the estimation of the parameter, then we use this branch, or you can say this techniques, estimation of parameter. And when the second opportunity is that we can construct a hypothesis, like we can frame that the population parameters is equal to this one, and based upon the sample observation we will test that whether that hypothesis is correct or not. So in this whole scenario, generally we have various methods, like we have various tests available with us. So why this today's session is relevant for us, because I am going to discuss a topic that is called Bayesian statistics, right? And I would like to discuss the application of this Bayesian statistics in clinical or biostatistics field. So how this topic is relevant for us, I would like to just give you a brief about that. So we already have discussed that we have a sample with us, and that sample is coming from a particular population, and population means that we have a particular objective regarding related to any particular study, and based upon that objective we define either we define a hypothesis or either we are interested to estimate the parameters, right? So in Bayesian statistics, what we usually do, we use all the techniques and tools that we already have discussed by when we talk about our frequency statistics. I will also come to that one, and we will compare that whatever the tools we already discussed in this frequency statistics, that will be applicable in again in this Bayesian statistics. But the difference is that we will get more, or you can say precise or good result as compared to frequency statistics. Now, the journey of this Bayesian statistics came from a author, or you can say a person that is called Thomas Bayes, who work in this area of probability. That means he was interested to see that how, when we talk about the probability of any two events, or when we talk about the probability of finding an probability of events, then how we assume that let us suppose A and B are two events. Generally we assume that they are independent, but the person Thomas Bayes provided us a theory that if those events are not independent, then how you will proceed further, right? So in that case, all of you are aware about a very famous theorem that is called Bayes theorem. Okay, he provided few results that how one can derive the probability when they are not independent. So if any event that happens before the you can say conducting an experiment or happening of the event of A, then how this event B impact the probability of happening of event A. Okay, so now let me discuss, or you can say first start our discussion with this author that is called Thomas Bayes. So Thomas Bayes, the person contributed a lot in the theory of this Bayesian statistics because he first developed the concept that how one event happen and that impact the probability of happening of the second event, right? So the thing is that this person never published his work. That means whatever the contribution or that is so, or you can say that whatever the current you can say updation we have in this Bayesian statistics, that is only due to his friend that is called Richard Price, because his friend you can say did some modification in the work that he already have done, and then he published his work. So the main contribution of this Richard Price came into picture when we talk about the Bayes theorem, because the work of this person Thomas Bayes, that was published by only by Richard Price. So let me first discuss about the this Bayes theorem. Then we will come to that how we uh, you can say correlate this Bayes theorem to the Bayesian statistics. I will come to that. So we already know that when we talk about a sample space, that means we have few events with us, and we generally consider a case that most of in our sample space we have mutually exclusive and exhaustive events. Okay, so I sometimes some person assumes that my talk is going to be very much mathematical, but I will try my best to keep it as simple as possible. Okay, so when we talk about a sample space and we have multiple events, so we consider those events that complete the sample space. So I'm assuming that this is my sample space, and I have certain events with me that are mutually exclusive and exhaustive. Okay, so it means, so when we consider the probability of any event A, so we are interested to find out the probability of any event A, and so it means how you will find out the probability. So we in our mathematics or in statistics and probability theory, we have a probability we have a result that is called total probability theorem. What is the concept of this total probability? That says that if we have any event A, so if you want to find out any event A, then that depends upon this you can say mutually exclusive and exhaustive events B1, B2, Bk, right? So you can write down them in this term: probability of A intersection B, and union of probability of A intersection B2, and so on, because you can see that there are few parts, or you can say the segment of this event A that lies over the common, or you can say the intersection of any event B come here, right? So that means if we consider that this is my second B2 and my B3, so you can observe that to find out probability of A, have to talk about that how this event A can be victims of other events B1, B2, Bk. Right? So the Bayes theorem came into picture. If someone event, we already have the probability or partition of sample space, and the probability of those events are non-zero, then how we can find out the reverse probability? So why I'm saying the reverse probability, because we already have that these are our partition of sample space, and the probability of them is non-zero, and then for any event A in S, that is called sample space. Okay, so probability of B are given A means that how you will find out the probability that we already have the information that A event has occurred, and we are interested to find the probability that it happens due to that it comes from the this you can say partition of this event, that is BR. So how you'll find it out? Okay, so you can see that, so it is already written here that for finding out the probability B are given A, what we are doing, we are finding first probability of A given BR. It means we first know that to find out the probability A given BR, so it means we have this partition A1 B1 B2 Bk, and how you will find out the probability of A given BR, that is already known to us. And we also know that how this probability of B given BR, right? Probability of BR, that how you will find it out, because we already have the information that these events are mutually exclusive and exhaustive, and the probability of them are known to us. So how you, in this way you can find out this probability. Now the thing that how we are going to generalize this concept in our Bayesian statistics. I will come to that later on. But the main thing that is very important for us to understand is that what we are doing, we are finding out the probability for the mutually exclusive event uh, by conditioning that already A events has happened. Okay. So let's suppose, so I'm going to give a brief more information about that. So we already have information that when any event, when we want to find out the intersection of any two events, the probability of A intersection B can be written as probability of A into probability of B given A. Okay, so we can arrange them and we can easily see that these are the mathematical representation, or you can say how you can write it down. But this is not, you can say, clearly understand if we not discuss this example. So I am going to discuss example with you that will help you to understand the concept. So example says, so FDI guidelines, according to FDI guidelines, so when we have any prior information given to us, okay, so prior information about topic that you wish to investigate in more details can be combined by using the Bayes theorem. So the information is that to find out the probability of A given B, when we already have the information of probability of B given A, and when we multiply this concept with probability of A and divided by probability of B, then you can find out this probability of B. So what is A given B? So this is called posterior probability. Why? The reason is that we have first have the information of probability of B, right? We have the information regarding probability of A, so that can be evaluated by total probability theorem that I already discussed with you. That we know that A is by using the total probability theorem, and we have the information of probability B given A, then you can find out a probability of A given B. So the likelihood probability of conditioning B being true when A occurs. So why it happens? So you can see that. So we have the prior information regarding this event A, okay, and and the evidence probability of conditioning B being true regardless of outcome of A. So here we are saying that here there is no concept of dependency of events B over the event A. So in this way, one can find out the probability of posterior. So what is posterior and what is likelihood and what is prior? So let me discuss it again in example. So in this example, we are talking about example of events of horse winning race. So we have the information with us that horse won 5 of the 12 races. So this is information that is given to us that among the 12 races, the horses won 5 races. But it had rained heavily before 3 of the 5 wins. So this is another information we have. That means that before 3 of the 5 wins, it has rained heavily. So this you have to keep this information in mind, that one race was lost when it had rained. So one race was lost when it has rained. What is the probability that horse will win the next race if it rains? So now you can see that. So here the we are saying that when it has rained, so in that case 3 of the 5 wins was registered, right? So what is the probability that horse will win? What is the probability that horse will win? You can say will win the next rain if it rains. So now it is so our main interest to see the probability of winning. We want to know the probability of winning it had rained. Okay, so what do you know? What we the information that we has is, so that information is we have is that it has rained, that probability of winning. So now you can see that it is the opposite case that we already if regarding the information that we already have. So we are dealing with we are interested to find a probability. What are the chances that horse will win the rain when that already rain has happened? Okay, so now the information already we have is that when the probability of winning when rain will happen, the rain is happening, that means 3 upon 5. So how you will get this? Because it is already given to us that it has rained heavily before 3 of the 5 wins. So now you can see that 3 upon 5 occurs, so that will give you probability 0.6, and the probability of winning is independent of the rain happen or not, so that is given as 5 upon 12. That is equal to 0.417, right? So now the probability of raining before a race, so it is given as 4 upon 12. So how you will get how you will see this information? Because If there is no rain happen, then how many wands that horse registered a wand, that is 4, and how many races among these one? 12, right? So from Bayes theorem we can see that this is our event, that is probability of, if we consider this as probability of A given B, okay, and this will be probability of B given A, and this is probability of winning is can be written as probability of this A, this can be probability of B. Okay, so we are referring this, we are, what we doing, we are implementing the result that we already have discussed in this theorem. So now why this result is important, why we not just consider that this probability of winning is just that this 5.12. The reason behind is that here, because raining impact the chances of that horse will win the race or not. Okay, so in our real life also, when we deal with any clinical data, or when we talk about any that information that related to the biostatistics, so we also know that when we try to test a drug over any human being, so we can't say that the impact of any drug on any human being remains same. Okay, because the person may be suffering from any particular disease, or you can say he's diagnosed or she is diagnosed with a particular disease. Then when someone is giving a particular type of drug for a particular disease, then the impact is different for one person to another. Okay, that's why Bayes theorem is very important, and that's why this Bayes theorem is generalized to the Bayesian statistics, and the concept is same. That means whatever the information we have, we will use that information, and that information is used in a broad manner. So here we are just talking about a few events. So we in the further we have converted this concept into this likelihood function, and then we have distributions with this. I'm going to cover them one by one, but the main objective is that, so if you want to find out any probability of any event for whom the prior information is given, but how to find out the probability that opposite of that event will happen then. So I have another example with me that will help you to understand the concept. So there are 3 manufacturers that supplies close to a retailer, right? So 60% of stocks come from the manufacturer one. So we have the information that among this total 100%, so 60% of the stocks coming from the manufacturer one, and 30% from the second manufacturer, and the remaining from the third manufacturer. So we have this information, and 10% of clothing from the manufacturer one is faulty. So we have this information with us that if the whatever the stock is coming from the manufacturer one, so 5% of the stock is this defective one, and from manufacturer 2, 15% is defective, and manufacturer 3, 50% is faulty from the third manufacturer, and 5% from the second manufacturer, and 10 from the first manufacturer. So now if we got a faulty item, so if you want to find out what is the probability that it is coming from the manufacturer 3, so what we are doing, so we are finding out the opposite probability. So let us suppose if we consider this the probability of each manufacturer as probability of B, and we are assuming that probability of A given B as you can say the faulty item that is coming from any of the manufacturers. We use A1, A2, A3 notation for that. But our main interest is that now if we got a faulty item, so if we want to verify whether this faulty item is coming from manufacturer 1 or 2 or 3, then this Bayes theorem plays very important role. Okay, so how you will solve this situation? So let us suppose let A be the event that government is faulty, and let BI be the event that the garment come from the manufacturer I. So now you can see that probability, we are interested to see that whether that government is faulty and it is coming from the third manufacturer. So it means we are interested to find out this probability, right? So now you can see that what we are doing. So we know that 50% of the items, whatever the item is produced by this third manufacturer, that is faulty, right? So this is faulty one, and we already have this information that 10% of the items is produced by this third manufacturer. So what we are doing, we are incorporating this information here, right? So here, so if we implement this concept here, so this will be probability of A given B3 into probability of B3 divided by probability of A. So what A here is? So probability of A means, that means the item is faulty. Okay. So now you can see that, so item is faulty can be come from manufacturer 1, can be come from manufacturer 2, and can be from manufacturer 3. So now see, you can see that after solving these probabilities, you will get this answer. So here, so we can see that manufacturer 3 produces only 10% of the garments, but needs 90% of the garments coming from this that manufacturer. Okay, faulty items. So now when we talk about when we talk about that, how many percentage of the faulty item is coming from this manufacturer third, without incorporating the any prior information, so it is coming as 50%. But when we talk about that whether we got a faulty item and what is the probability that it is coming from the third one, so the probability has changed now. Okay, so it means we are getting more accurate information about this whether about the performance of the manufacturer. Okay, so now before so if you want to see from this you can say tree diagram, that is also helpful to understand the concept. So we have seen that we have three branches that shows the percentage of the items coming from each manufacturer, right? 60%, 30%, 10%. And after that, you also observe that if we extend these branches, so you can see that 10% shows that if item is coming faulty from the manufacturer 1, 5% item coming from faulty from the manufacturer 2, and so on. 50% is coming as from the third manufacturer. So if you want to find out the probability that if we got a faulty item, what is the probability that it is coming from the third manufacturer? So now you can see, you can directly multiply this concept and easily find it out that if we want to find out the item is faulty and it is coming from a third manufacturer, so we have these probabilities, right? So it is coming at 0.015, right? And we also know that what this probability should come from the that how many that what is the probability that the item is faulty. So when you divide this concept by this one, then you will get the same answer that we already have discussed. So this is the base of the you can say Bayesian statistic that we are going to discuss now. So first that I have discussed these simple examples with you. The reason is that so you can, if you can understand the concept from the probability theory, then only you are able to understand the concept from the distribution point of view. Okay. Now I have another example. Let me say that people. So the probability is 0.001, for which there is a diagnostic test which is 99% effective. Okay, so it means we have a diagnostic test that is 90% effective. A random subject takes the test. So a random observation, that means a random person takes the test which says positive. So it means if any person take the test and it is positive, when that means the person has a rare disease. Okay, so what is the probability that they have X? So now what do you want? Now you can see that, so 0.992, 0.01, what does it shows? It shows that probability of A intersection B, or you can also write it down as probability of A given B into probability of B. And if we are interested to find out this probability of B given A, so these are the things. So what is your probability of A given B? That shows that if any person got a rare disease with this probability, then what are the chances that probability that it has that disease? So you can see that this is 0.0902. Okay, so before going to discuss further, I would like to introduction about the clinical trials, because my main objective is in this talk is that I want to introduce the concept of Bayesian statistics in a clinical field. Okay, so there are a few very basic things and concept that we have to discuss in this session, that is called that what are the few necessary and concept and key components that are essential to clinical trial design. The first is called protocol. Okay, so in protocol we discuss each and everything, we define each and everything what we incorporate in our study, and what will be our subject and how we will conduct the study. And after that we have primary objectives. So that shows that why we are conducting a study. Each and everything is defined clearly. And eligibility criteria shows that what which persons are, what will be our population. And then we talk about the power and sample size, how we will define our sample size, how we will define the power of our study. Then we have blinding. So basically in clinical trial, when we conduct any study to get more accurate result, so we use this concept that is called blinding. So it means how we assign our, you can say, treatment to the patients, then how whether to make it randomized. So it means that, so let us suppose we have you can say drugs with us, we have some medicine with us, and we want to you can say give those drugs to the patient. So we do not uh, to make it more you can say clear that we do not making any error. So that's why we use this concept. So randomization is also helpful for that. So in which uh, we can say that we are applying tools that in that way, such that we got assign the observe, or you can say we assign our medicines to the person randomly. That means there should be no any systematic error or biasness there. So then we have parallel and crossover design. So parallel means when we conducting two study parallelly, and crossover designs means when we have what we're doing, we have two designs, and after some time period, whatever the patients we have in the first design, we will change them. So then we talk about the data collection, that how we can collect the data set, and adverse events we will also talk about them, that what will be the adverse event in our clinical trials. Then we talk about the closeout, then how we close out the study. And then we have pharmacokinetics, and then pharmacodynamics, and then we have different phases. So first I will discuss about what is protocol, and that is very important. So protocol is a document that provides a comprehensive description of the entire study. So it means, so if you want, if from the protocol you will get a clear understanding that what we will do in our study. Okay. So it includes the general information and background of the disease or condition to be treated, the intervention to be used, the study objectives, and statistical design and methodology. So each and everything is written clearly in the protocol. So the protocol should also describe the known and potential risk and the benefit of the treatment to the human subjects. And then we have the study protocol that may be viewed as a written agreement between the investigator and the trial participant and the scientific community at large. So let us suppose if someone wants to conduct a study, someone wants to collect a sample and wants to visit the field, then one has to prepare, you can say, one has to keep the information of the respondent, that is not to be disclosed with anyone. So one has to clearly mention that this study is for the development of the drug, and for the improvement of the drug. So that's why the person or the respondent agrees to give his feedback. Otherwise he or she will not disclose its responses, or you can say personal information with the, you can say, investigator. So the study protocol may be viewed as a written agreement between the investigator, the trial participant, and the scientific community at large. It is a single document that specifies the detailed research plan and lays out the systematic and organization for the trial. So now you can see further that in our protocol we have these 8 segments. And in the first we have introduction and rationale. Then we have study objective, including primary and secondary aims. So in the study it is possible that we can have a single aim, and we can have more than one aim. So overall study design and experimental plan. So in the next we have overall study design, that means how we will conduct our study. Then we also discuss about our experimental plan also. Then we have patient population and then enrollment criteria. So how we will choose our population, then what will be the enrollment criteria for them. Then we have specific treatment procedure such as randomization, intervention, outcome measurement, and possible protocol deviation. So it may possible that during this experimental stage we find that whatever the things it is written in the protocol, so how much deviated our study will be there. So we have to mention each and everything, that what are the possible in real scenario when we conduct a study, that there is a possibility of deviation. So we will give a little bit information about that. So then we talk about the statistical consideration. So which type of result we will get and how we will analyze that. Then we talk about safety data collection and reporting. Then regulatory administration and legal obligation. We will discuss each and everything. So let us suppose if our study is helpful for the development of the drug, then each and everything legal obligation we also take about consideration. Okay, next we have primary objective. So as already discussed earlier, that in our protocol we may define either a single aim or a multiple aims. So first we will so our main aim should be, so we will first focus on our main aim, that is primary aim. In general, a clinical trials contain one primary objective and possible several secondary objectives. So all the objective must be feasible. So it means that they must be attainable and clinically meaningful. So that means if someone is conducting a study, that must have a meaning. So the primary objective of a clinical trial is the main question that an investigator are more interested in, and the trial should be able to adequately address at its conclusion. For example, so I will also discuss what is phase 1. So in a phase 1 trial for a patient with acute leukemia, the primary objective was to determine the maximum tolerated dose and the associated dose schedule for a new drug. So in this way, so our objective should be clear. Then only we can conduct a study. If we have you can say not clear objective, that will be not beneficial to conduct the study. Then the next stage says that eligibility criteria and accrual. The study population is subset of the general population. We already know that, because we can't reach each and every person to complete our study. We have to take only sample. So which should be defined in advance with unambiguous inclusion and eligibility criteria. So we must take into consideration the impact that these criteria will have on the study. So it means if we are taking a particular population, or you can say a population from a particular region, so we must be aware about that this population, how this population, or you can say responses, will impact our study. So whether it is feasible or not, and eligibility criteria tied with the patient safety and the anticipated effect of the intervention. So I'm going to discuss example here. So let us suppose if we are giving a particular drug to a patient, then you must be take care of the patient safety from the point of the side effect of the drug. And you also talk about the participant defined in the inclusion criteria of a trial should be those who have the potential to benefit from the intervention and whose are likely to be adhere to the study protocol. So it means, so whenever we testing a drug either on the patient or the you can say participant, so uh, you must have a clear idea that this drug will be beneficial for them. So patient who may be harmed by the intervention must be excluded. So you have to you can note down each and everything, that when we have to exclude the patient, or you can say when we have to exclude a respondent, and those at a high risk of developing condition. So it means you, while conducting a study, you should not, you should talk about a clear you can say prior history from the patients. Then only we have to take those observations in our study. Then we have a power and sample size. So basically when we talk about the power, so why this thing is important from the point of statistical thing, the reason is that how whatever the statistical tool you are using, so how much it is powerful to detect that shift. So basically when we talk about sample size, so that also impact the power. So generally we consider that by taking a large sample we will get more accurate result. So for that, we also talk about important thing that is called level of significance. So level of significance is basically a value that shows that how much uh, error we are accepting in our population. So generally we consider that 5% level of significance is assumed. So it means it shows that we are assuming that that 5% type 1 error will be there. Okay, so it shows that while conducting a study, so we generally assume that 5% error will be there, right? So level of significance and power are essential part of the clinical trial planning. So in that case, the design parameter such as the effect size. So it means n is called sample size. So how much samples we have to take, that depends upon the level of significance. That means how much errors we are considering in our study. And we also talk about the epsilon, that is called error, or you can say tolerance level. So that determine the this n value, that is sample size. So our whole study, you can say if we are finding this n, that is called sample size, so that depends upon level of significance, that depends upon the epsilon, that is called tolerance level. So based upon that, one can define that how much sample size we should consider in our study. The design parameters such as the effect size included the expected treatment difference and the associated variance. So these are the another things, or you can say that we should consider by determining the sample size and other things. So clinical trial should control the type 1 error. So type 1 error is called, so when we are rejecting something and when actually it is true. So if we from the clinical trials, so type 1 error means, so let us suppose you have a drug with you, and from the, you can say, responses from the patient and from the observation, you find that based upon the statistical test, you are rejecting that this drug is not beneficial for the patient, but actually it is beneficial. Okay, so it means we are conducting an error. So it means prevent false positive results, and we should have sufficient statistical power to detect the treatment difference considered to be of clinical interest. So statistical power means, so let us suppose, generally we talk about a simple terms, then let us suppose we have frame a hypothesis that mu is equal to mu naught, and alternative is mu is not equal to mu naught. So how much our test is powerful to detect a shift? So let us suppose sometimes we have various tests for a single testing, but when we talk about this statistical, you can say, power of a test. So how much power how much power your test has to detect a single shift? That means your me is, let us suppose, 5. So whether your test is able to detect the shift that it is not equal to 5 or not, right? So this is the power of the test. So, and it is also important that you should clearly define which statistical test you are going to do. So the next step is called blinding. So I already told you that while conducting a study, we want that our studies is conducted without any error or biasness, so that there are three ways to remove the biasness, right? So that is the first way is called a single blind study, so in which patients are not aware of which intervention they receive, but the investigator are. So let us suppose we have two groups, X and Y, and uh, and we have two treatments, A and B. So A can be placebo, B can be actual treatment. Okay, so in this case of blindness, the patients that are in the group X and those are in the group Y, they do not know whether they are getting a placebo or whether they are getting the actual drug. Okay, so that is called single blind study. So in a double blind study, but in the single blind study, the difference is that, but the investigator know that which group has received a placebo and which group has received the treatment. In a single blind study, neither the patient nor the investigator who are responsible for the for patient follow-up know the identity of the treatment. So in that case, in the double blind study, you can see that the investigator also not aware about, so which group got the placebo or which group got the treatment. So in this case, so they are conducting a double blind study. So in the triple blind study, not only this patient investigator are unaware about the treatment assignment, so but also the person who is doing this, who is conducting the data analysis part, because sometime you can easily see that, so while conducting a study, or you can say while doing the analysis part, note down the values in terms of numbers 1, 2, 3, and so on, so that we can conduct our study. But and sometimes we used to write the names of the patients, and we also have the information with us that this group belongs to this placebo and this group belongs to the treatment. But so this information is known to the, you can say, the person who is doing the analysis part. But in this triple blind study, so those so they did not mention that which value belongs to the placebo and which value belongs to the treatment. So they again code it. And so in this triple blind study, neither the patient, neither the investigator, neither the person who doing the data analysis part know each and everything. Okay. So next, we have randomization. Already discussed with you that randomization play very important role, and why it is important. The reason behind is that it removes systematic error or biasness, and it is considered as a foundation of statistical analysis. So why randomization is so important? The reason is that, so let us suppose we have a few observations with us in this group X and in this group Y. So if we have prior information about that the members who is group A, X, and who is those in group Y. So if we are not assigning the treatment to randomly to this X and Y, then you can easily see that your result is one-sided. Okay. So for example, if the physician always assigns healthier or this younger patient to the experimental therapy and sick or old patient to be standard therapy, the study may eventually conclude that the experimental therapy is more effective because it is a one-sided case. For that, it is important that randomization should be there, and that also reduce, or you can say, any biasness and any type of error. Okay, so I already discussed with you that we have two type of design. One is called parallel and second is called crossover design. So what is the difference between these two designs? The difference is that in the parallel design, we are conducting two studies simultaneously. So from this graphical representation, it is very much clear to you. So let us suppose we have two treatment with us. One is treatment A and second treatment B. And simultaneously, we are conducting a study in which we are taking the effect of treatment A and treatment B. And here we are taking different patients, right? So it means we are conducting two studies simultaneously. Okay, in the first study, you can see that we are taking few patients and whom we are giving treatment A, and in the second we are taking few patients and for those treatment B is allotted, right? So similarly, we have different patients from and we are giving treatment A and treatment B to them. So the difference is that, so this is called parallel because we are simultaneously conducting two designs. So in the crossover design, you can see that, so after, so here what those patient who has received treatment A, now they will be given treatment B, and those patient who are in treatment B, they will be given treatment A. So to normalize the effect, so what they usually do, they consider the concept of washout period to reduce carryover effect. So let us suppose, so they waited or they put a gap of 10 or 15 days or 30 days, so that, so the patient who received treatment B, so the impact of any treatment B will be negligible, then only the patient receive now treatment A. So this is called crossover design. So in this way, they will try to see the impact of whether actually the effect of this treatment A and B is coming or due to natural, this you can say random or chance. So this is the concept of this parallel and crossover design. So the next thing that we have is that is called data collection. So the clinical trials data are collected through medical examination. So that is very much clear. So either you can collect the data through medical examination or through laboratory test, then interviews and questionnaire. We need to devote sufficient effort to ensure that data are critical to the interpretation and conclusion of the trial of the high quality. The reason behind is that because if you are noting down the values through medical examination and laboratory and other things, so while noting down, so there are possibility that we commit a mistake in noting down, right? So we have to first find out those errors before doing any analysis. And sometimes it happens that noting down the weight of any person, so one person is saying that the weight is 78, but other person has written the weight of the person as 87. So these things already happen. So it is essential that, and important, so we have to also rectify or you can say we also have to you can say uh, cross check whether all the observations are recorded properly or not. So response variables are the outcome measured in the course of trial. They must be clinically relevant and also must define and answer the primary and secondary question. Because why this is important for this you can say for our objective. The reason is that because based upon these responses or whatever the you can say data set we have, based upon that we after doing the statistical analysis we whatever the result we got we and those results are correspond to this our primary and secondary question, because we our main interest to see that whatever the aim we have, so whether our result is coming according to that or not. Okay, the next thing that we have is adverse event. So while conducting at clinical trials, we also observe this type of study, or you can say we also observe these types of event or cases in which we also see that there are few side effects appear. Side effects are always on the top of the concern for patient and investigator. Toxicities must be continuously monitored throughout the trial. The reason behind is that, so let us suppose you are giving a particular type of drug to the patient, and if we keep repeating, or you can say you are again giving the same drug or different type of combination of drug to the patient, so that may lead to the toxicities level increase in the body of the patient. So the potential benefit of the intervention should be maximized. So while possible toxicities should be kept to be minimum by the appropriately selecting the drug. So it means while observing the impact of a particular drug, our main objective is that there should be less negative impact of the that drug on the patient. And at any time, a trial should be suspended if excessive toxicities have occurred. So it means we should stop the you can say intervention if there is such cases. So next thing we have is called closeout. So it is very important. Why? The reason is that, so because when we stop conducting our this clinical trial, the close of a clinical trial usually require careful planning, such as consideration of the date when the last participant is enrolled and the minimum length of the follow-up period. Okay, so after completion of the trial, it is important to clean and verify the data. The reason behind is because now if you have all the information with you, now the next step is to clean and verify data, and then we do the statistical analysis. Okay, misspecified models and inappropriate models methods may result in misleading findings and impair the credibility of the trial, because sometimes it happens that we have lot of tools and techniques available with us, and without you can say validating whether the assumptions of the model or whether our data set satisfy those assumptions, we apply those tools over the data on the data set, and we got you can say wrong results, or we got inappropriate result, but some persons use those results and come up with a result that this is the final one. So these are the few possibilities we have to take care of. So questions as follows may be asked at the conclusion of the trial. Has the trial been carried out as originally planned? So this is the main thing. So where you have to cross check whether the trials, whatever that is mentioned in the your protocol or your aim or objective. So whether you are able to achieve that objective or not. How do the result of the trial compare with those from other relevant study? So you can also compare and see so whether the output you are getting, whether it is you can say compatible with them or not. So what are the clinical implications of the study findings, and how will the trial conclusion affect clinical practice? So this is very important thing. So sometimes we got such type of results, so we have to take care of that. And in the closeout you can see that, so when there is a very simple concept that can be easily understand. So you can see, so when a person is given a particular type of drug, so after, with the passage of time, so you can see the impact of the drug keeps on decreasing. Okay, and why it is important? So this is the reason behind is that we are going to discuss this concept, and this is called pharmacokinetics and pharmacodynamics. The reason behind is because using this theory, one can easily see the impact of the drug and what is the time duration for that, and one can also observe that what you can say uh, which amount of or the tendency of the drug and the quantity of drug and what quantity we should recommend to a particular type of patient. For example, so in the clinical studies and early phase trials, we need to understand the pharmacokinetics and thermodynamics of the drug. So PK works, that is pharmacokinetics, works on the kinetics of a drug consumption, which can be easily observed that CT that denotes the drug plasma, and D is the dose of the drug, and V is the volume of the distribution. So B is the elimination of the rate. So that can be easily be observed by easily be understandable. So now you can see that. So at the starting stage, that means at the starting stage we observed that the time period is 0, and at that time any person consume or has taken a drug. So you can see that with the passage of time, so there is a tendency that that drug dissolves in our blood and the impact is coming. So now you can see that here, that with the passage of time, the drug concentration in our blood keeps on increasing, and after a particular time period, the impact keeps on decreasing, and that's why you can see that after a particular time period, now the impact of drug is coming to be close to 0. Okay, so pharmacokinetics study about this thing, that how many hours a particular drug will take to you can say give its impact, and how many hours it will take to dissolve it completely. Okay, the next thing is called thermodynamics. So what is the role of this thermodynamics? So that can be easily be understand by using this by this graphical representation. So this from this graph you can see that we are taking drug concentration on the x-axis, and we are observing the effect of the drug on the y-axis, right? So this is called Emax and sigmoid curve. This sigmoid curve shows that with increment in the drug concentration, so when you increase the drug concentration from 0 to 12, and there will be an increment in the effect also. So it means that, so how much quantity of this drug concentration we have to increase so that we will get a complete impact. So that can be easily observed, and this vary from one drug to another drug. Okay, so here next concept that I'm going to discuss with you, that is called phase 1 to phase 4 of clinical trials. These are very important because as we already discussed that in the protocol, each and everything is defined clearly, that how we will conduct the study. So in this phase 1, our main objective is to see that the first step of testing a new drug in human beings is to understand and how well the agent can be tolerated. So this is the thing that is important, that how we will test a new drug in human beings and how will the agent can be tolerated. The safety of drug does not mean absolutely harmless, because we already have observed that consumption of drug will increase the toxicity level in the blood. So we also have to observe that how much negative impact from the drug that is coming in the body of the patient. So while it is often necessary to accept a certain amount of risk, a drug may be considered safe as long as it benefited outweighed the associated risk. So let us suppose if there are patient suffering from heart disease and other you can critical disease. So if you are giving a very high quantity of high amount of steroids and other things, so that will reduce the impact from this critical disease, but it may have some serious effect also. So for example, one can easily understand that chemotherapies are known to potentially induce very severe adverse effect, but that will save the life of the patient. We already know that. So however, they have some negative impact, but it is essential it is essential that they have to use the chemotherapies, because uh, although it has negative effect, but it saves the lives of the patient, right? So this is the case that we have to consider in the phase 1. Now we have in the phase 2, we have discuss about the trials in which proof of the principal stage of drug development. That means you observe that how one can construct the drug for a particular type of disease. This phase of trial evaluates whether a drug has any biological activity or beneficial effect. So after implementing, or you can say after observing the value from the phase 1, now we can decide that whether we can proceed further with this drug or not, because whether it is actually beneficial for the patients or not. Okay, so and it continues. Okay. So phase two studies provides critical information for the go or no go decision, because whether we should continue with our study or not. So that this discussion came in this phase two. So that is whether to proceed further to large confirmation phase three trials. So non-working drug should be dropped from the study or killed at this stage to avoid investing more resources and efforts. So phase two is very important, and the phase two trials may be single arm, assessing one treatment, or multi-arm, comparing several treatment. So single arm means what we are doing in this phase two trial, either we are taking one drug at a time, or either we are conducting the same study for different treatments also. Okay, multi-arm, so in which we are taking several drugs at a single time and observe the overall effect. So randomized phase trial may reduce the so-called trial effect, and which often arise due to the different patient population, physician cares, and medical environment between the current and previous studies. So here next stage is called phase 3. So the phase 3 trials are large scale in terms of resources, because now what we're doing, if our drug is you can say acceptable in the phase 2, then only we conduct the phase 3 trial. Otherwise we stop there, and we will never continue with that study. So this phase collects a large amount of data over a long period of follow-up to examine the ultimate therapeutic effect of a new drug. The standard form of a phase 3 trial is a double-blinded. Already discussed with you what is double-blinded. So in which neither the patient nor the investigator know about this whether the patients are getting a particular which type of drug they are getting. So a randomized one, and the placebo control study. Placebo control study means we have two groups with us, and one group is given a placebo, that is a particular type of medicine in which there will be no salt, and control study in which we are dealing with a group in which they are giving a particular type of drug. So the control arm may be placebo or the standard of care. The use of a placebo in a randomized trial is only acceptable if there is no other best or standard therapy available. So that is very much clear to you. So now coming to the this phase 4. So after a drug successfully passes through phase 1, 2, and 3, so it means in the phase 1 we observe the impact of the drug on the patients. Then in the phase 2 we decide whether we will continue with that particular drug and whether we can introduce that drug to the in the market or not. And in the phase 3, if that drug is acceptable in the phase 2, then we will come to the phase 3, in which we conduct a very large study and we get all the information regarding from the patients, and we will do statistical analysis of that. And now the in the phase 4, we will do, we are more concerned about that our drug will get approval. So a phase 4 trial may be initiated as a post marketing surveillance study. So in this phase, the drug associated adverse events and patient safety are closely monitored. So it means we will observe that what will be the side effect of those drugs to identify problems that have not been recognized prior to this approval. So rare severe adverse events, we will also see that what are the side effects of those that arise after the widespread use of the drug in the general patient population may add a warning label to the prescription. So it is already written sometime on this on the medicine cover that this drug is not meant for diabetes patient, this drug is not beneficial for a particular type of patient. So in this phase we note down each and everything regarding a particular drug. So a phase 4 trial is an integral part of the drug evaluation process, which may lead to new indications or new uses of the approved agent. So this is the you can say graphical this flow chart way that one can easily understand from this flow chart, that at the initial state we have a trial design in which we plan that how will we conduct the study. Then we also have this protocol review, and in that case you can see that there is an internal review board that observe that whether each and everything is conducted properly or not. Then we move to the next thing, that is called trial open, and trial conduct interim monitoring analysis. So in that case, Day-3 SATA monitoring board will take care of the information that we have gathered. And if it is considered satisfactory, then we move to the next one and the trial will be closed. Okay, so it means in the clinical trial, our whole aim is to eliminate the bias. Whatever the study we conduct, that should be unbiased, that should quantify and reduce errors. So produce accurate estimates of treatment effect, and that we will see that what are the association between the variables. We will also see observe that, and our main objective is to provide a high degree of credibility, reproducibility, and internal and external validity. So it means that, so whatever the conclusion we will get from our study, that should be of high credibility. That is very important. And we will reproduce, if someone wants to reproduce the same thing, that one can easily reproduce that. And that should be internal and external validity. And that, and most importantly, influence future clinical practice. The reason is that, so if someone has you can say develop a new drug and that is beneficial for the patients, then how one can improve, one can introduce a more improved drug that will be helpful. So if that person observe all the, you can say, drawbacks and positive points of that particular drug, then only he or she can produce or develop the drug, right? So how this this now I'm coming to the this we already have discussed a lot about the clinical trials and what are the steps that incorporate in the clinical trial. Now I will discuss with you that how we will implement the Bayesian statistics in clinical trial. So that is very important. So I already told you that in the Bayesian statistics, so we consider that any prior information that we have regarding to any particular you can say event or not event, that will change the you can say that will impact the final probability, right? So in that case, there are, so this study was very developed earlier, and in 1950 lot of people are working on this theory. Okay, and there are a lot of tools available, and but the reason behind is that why it is not you can say famous earlier. The reason is that because it is very complicated. So let us suppose if we are only dealing with this probability of event A, then it is simple to find out. So to find out the probability of A, we will our main concept is that to find out the favorable event regarding to this event A divided by the favorable event total events that we have in the sample space. Okay, but when we talk about this concept, that is we are interested to find the probability of event B given that A already has occurred, then it is very difficult, because if we do not have the information about probability of A given B, or you can say probability of A intersection B and probability of A, then it will be not easy to find it out. Okay, so how one has generalized this result? For that, there are a lot of research is going on nowadays. And with the help of computer and other methods, so now we use you can say numerical approximation tools and other things that help us to solve these complex cases. So here we can see that, so Bayesian statistics is very important in the designing and analysis of the clinical trials, and these are the authors Cornfield and Friedman, and at various higher you can say very well-known institution, National Institute of Health and this UK Medical Research Council. They use Bayesian statistics a lot, and till the time 1990, and by 1995 the Bayesian statistics become, you can say, very popular. The reason behind is that due to the computing power. Nowadays we can use Markov chain multi-Carlo method and other techniques with us that helps to, you can solve complex problems. So now here I'm going to first discuss with you the few you can say distributions, because what I'm doing now, I'm, in spite of taking probability, so I'm considering distribution in place of probability, and then I will show that how one can find out the Bayesian statistics, or you can say that is called posterior distributions also. So first of all, we must know about that what is distribution. I think if there are few persons who are not aware about the distribution, I'm just giving the brief about that. So when we talk about a distribution, so it means we're talking about a function, mathematical function that satisfies some assumptions. Okay, So if we consider any x as a random variable and we define a function x and we call that function, if that satisfies few assumptions like it lies but it values lies between 0 to 1, when you take summation if it is a discrete one, and when we take integration over the range of this function, then it is coming as 1, and this is this cumulative distribution function and it should be right continuous. So if we our if there is any function that satisfy all the assumptions, such type of assumption, then we call that as a distribution. Okay, and there are a lot of distributions available in the literature, and I'm going to discuss few of them. The reason behind is that why it is essential to consider distribution in place of the single this probability concept. The reason is that there are a few situations where we can model mathematical functions over the data set. For example, let us suppose we have such type of data set. When we construct a histogram over the data set, you can see that the shape of the curve is of such type, and we generally say that data is of data the shape of the data is bell-shaped curve, then we can use normal distribution in the case. So there are few cases I'm going to discuss that will help you understand. So the first distribution is called discrete uniform distribution. So discrete uniform distribution means that we are dealing with a discrete random number, and here we are saying that whatever the numbers we have, so here we can see that we have k numbers and we are giving equal weight to them. So here, so where x1 x2 are different values on the real line and k is positive number, the discrete uniform distribution is often used in the case of context of Bayesian model. So when we have such cases in which we want to assign equal probability to to our you can say variable values, specific values, and we want to find out the probability. So it means we are giving equal weights. So simple example one can easily understand from the experiment of conducting a coin or conducting or if you can say conducting an experiment for a coin, like flipping a coin or rolling a dice. So you can see that while flipping a coin, we have two possible outcomes, that is head or tail, and we usually assign equal weights to them. Okay, and when we used to roll a die, we have 6 possibilities, so we assign equal probability to them. So this is the same case here. So it means what are the possible values we have, we are assigning equal weights to them. So that's why we have this probability 1 upon k here. So why this is important in our Bayesian analysis? The reason is that sometimes there are situations like that we are not aware about any prior information about any particular. Hello. So we are not aware about any prior information about the distribution. So it means why? So in that case, when we have no any prior information about any event, so this is considered as a, you can say first choice to consider this as a prior distribution. I will come to that what is prior. So let me first introduce with you few distributions, then we will come to that. So the next is called Banali distribution. So this distribution is very, I think most of you are aware about this distribution. The reason is that this is most widely used when we have two outcomes. One is 0 and other is 1, and we want to find out the probability. So this is the mathematical function of this distribution, Banali distribution, and it is widely used in the case of phase 1 and phase 2. So what is phase 1? In which we observe whether the drug is effective for the patient or not, and whether it is beneficial. So in that phase 2 we draw the conclusion whether the drug is actually effective or not. So in which binary endpoints are typically observed. For example, so whether a person had experienced toxicity or not, so whether a patient has responded to the treatment or not. So it means either the patient responded, then we usually assign the value 1. When the patient is not responded to the treatment, then we usually assign value 0. So in that case, it is better to use this distribution when we conducting a single trial in which we are only interested to see that whether it is take value or 0 or 1. So in that case, this Banali distribution is considered as a good one. The next distribution is called binomial distribution, and if So basically, binomial distribution is a generalization of Banali distribution. The reason is that, so in the Banali distribution, we have observed that variables, if we consider x as a discrete variable and it takes only value 0 and 1. So when we conduct, and it is this is the outcome of a single experiment. That means we are observing this value for a single patient, and when we have lot of patients with us, and those patients are considered as independent, okay, and they are identical distributed. So binomial distribution can be used to find out the probability in that case. So the function is defined this way, that it is given as ncy py 1 minus pn minus y. So the binomial distribution may be used to model the number of patients who have experienced toxicity. So now in the previous case, we have discussed that the patient has toxicity or not. So this is, so we are talking about only single patient. Now we are talking about that how that we want to model that whether the number of patients. So it can be n patients. That's why we are observing that n value here. That means we have n patient, and we want to see that how many of how many patient among this n patient experience toxicity, whether they responded to this treatment or not. The next thing that we have is that is called beta binomial distribution. That is, you can say, mixture of two distributions. One is beta and second one is binomial. Okay, so basically beta is a continuous distribution. So when we call any distribution to be discrete or continuous, so let us suppose we have x variable with us, and if it takes a discrete value, so that means 0 1 2 and so on. Okay, and when we talk about any variable x that takes value in the interval, or when so that is called the continuous one, and the when we it takes value 1 2 and 3, so it means a complete whole number. Okay, all a whole integer value. That's why it is called discrete variable, and it is called continuous variable when it takes value in the interval. So it means it incorporates all the values that lies in this 0 and 1. Okay, so beta binomial distribution is a combination of discrete and continuous distribution. Okay, so in that case you can see that the function is defined as a discrete variable, that is x is a discrete variable, although beta is a continuous variable, but the combination, this beta and binomial distribution, will again be a discrete distribution. So this, the form of the PDF is given here, and you can see that the mean and variance is given. So why it is going to be important, I will discuss in with an example here, that how you will get a beta binomial distribution. So when we talk about normal distribution, it's most widely used. I've already discussed an example with you, that let us suppose we have any data set that while constructing a histogram, we observe that the plot of this frequency curve of the data is of such type of curve, right? So we usually assume that data is coming from the normal distribution. So the PDF of the normal distribution is this one of 1 upon 2 pi sigma exponential minus x minus mu whole square upon 2 sigma. And why it is important? The reason is that, so let us suppose when we have a very large data set, so it is easily be observed that that data set can be approximated as a normal distribution because of this property of central tendency theorem that says that for large data set you can assume that that standardized form of any variable for this converts to the normal distribution. So the next distribution that is called gamma distribution. This is the mathematical form of this gamma distribution, but why it is important? First, let me introduce the concept of this gamma distribution. So basically, gamma distribution came from a very well-known distribution that is called exponential. Okay, and exponential distribution is basically used to study, you can say, lifetime of the components, or to observe the survival of any survival of human beings and living organs. Okay, so when we talk about gamma distribution, so that is coming from the generalization of exponential distribution. So let us suppose x variable that follow exponential distribution is some parameter lambda. Okay, so when we have two such variables x1 x2 and both are independent, so that means when we want to find out the distribution of x1 plus x2, so that will become this gamma distribution, and the form of the gamma distribution here will be alpha will be 2 here and beta is equal to lambda here. But why it is important? The importance comes from the modeling of the hazard function. So what is hazard function? So in survival analysis, our main concern is that what is the probability that what is the probability that any living person, any human being who survive a particular time period. So let us suppose we have this line that shows that means that this value shows a newborn baby, and this is the this value of x shows that person already has attained a age of x, and what is the probability that he will die in the next 1 hour, and what is the probability that he will die in the next 5 minutes. So sometime we are more interested in that. So in that case, gamma distribution is oftenly used to model such type of cases, because so when we say the modeling to model something, the reason behind is that, so that depends upon the shape of the data set. So let us suppose when we have any data set that is of positive skewness, and when we have any data set that is of symmetric shape, and when we have any data set that is of negative skew, so we have to choose such distribution that has the same characteristics. So in this survival analysis, if we are interested to observe such cases where the shape of the gamma distribution is such type that can be you can say fit the data set of such type of shape. So that's why it is very helpful. So the next distribution that we have is inverse gamma, and the reason behind is why we already discussed the case of gamma distribution, then why there is requirement of this inverse gamma. The reason behind is that there is a only difference of the gamma distribution and inverse gamma distribution is that here the gamma distribution we observe that x follow gamma distribution, but in the inverse gamma we consider 1 upon x follow this gamma distribution. So that's why y follow inverse, why it is called y follow inverse gamma. Okay, so there is a little bit variation in the PDM. The reason behind that why we consider inverse gamma as a for this study, the reason is that because it is considered as a very good distribution for a prior conditions. For example, when we are talking about this x variable that follow normal distribution, so we have two parameters with us, mu and sigma square, right? So mu represents the mean and sigma square represents the variance. So generally, how we will determine this value mu and sigma square? We have some observation with us and we estimate these values of mu and sigma square from the sample. But what happens if these mu's are not a constant? So generally we assume our parameters as a constant. So what will be the case when these parameters are not constant? They are again a random variable. So we if we assume that sigma square that denotes the variance of the normal distribution, it is a not constant, it is again a random variable, then so it is generally assumed that sigma square follow inverse gamma. So that means the variability of this the variability parameter of the x variable that follow again another distribution, and that distribution is inverse gamma. So that's why it is important to study this inverse gamma distribution. Then we have Weibull distribution. So it is also widely used. The reason behind is it is that it has certain characteristics like flexibility of the survival function. So the importance of this Weibull distribution appears from this expression. Okay, that Weibull distribution of this has this PDF of this form, and it shows that when we this is called shape parameter alpha, and the property says that so when we put alpha greater than 1, so it has a different characteristics. When we consider alpha as less than 1, that has different characteristics. For example, so let us suppose we are dealing with such type of study in which we want to see you can say the death of human beings by consuming a particular type of drug. So if we model that situation with the Weibull distribution, and from the data set we got the value that alpha is coming something 1 point something. Okay, so it shows that the person has a higher tendency of you can say or probability of the dying the person is very high. Okay, so if from the data set we got the value that alpha is less than 1, so it shows that hazard rate is coming to be decreasing decreasing trend. So it shows that the effect of the drug with the time decreases. So in the first case when alpha is greater than 1, so it shows that with the passage of time that chances of the drug you can say the negative impact of the drug is coming to be more as compared to when alpha is less than 1, the impact of the drug decreases with time. So when we consider alpha is equal to 1, so that will be equal to the this exponential distribution. So this is, so all three distribution, exponential distribution, gamma distribution and variable distribution, they are related to each other, and they gamma distribution and variable distribution, they are both, you can say, well from the exponential distribution, so they are called generalized case of exponential distribution. Okay, the reason behind why we are discussing these distribution, this is very important for understanding the concept of Bayesian statistics. So I already discussed with you at the starting of this session that we generally use frequentistic method. What is this method? This method shows that, so let us suppose we have few observations with us, x1, x2, xn. Okay, and our main objective is to find or inference you can say extract information. So what these observations has, right? Let us suppose we generally assume that this data set follow any distribution like normal with some parameter mu and sigma square, and we estimate the value of mu and sigma square from this observation, and based upon that we can say that our data set is of our data can be modeled by using a particular distribution of normal by with these parameters mu and sigma square. But how this different, how this Bayesian statistics different from this approach? The reason is that in our Bayesian statistics, we consider that these parameters mu and sigma square, they are not constant. Okay, we in our study we assume that these parameters mu and sigma square, they are again coming from they again follow a particular distribution, and we are taking a specific value of mu and sigma square in this case, then only we can say that x follow normal distribution. For example, I will come to that. So first, to understand the concept of this frequentistic method, we first have to discuss this theory that is called maximum likelihood estimation, that says that let us suppose we have our y variable that follow particular distribution that has this PDF of this form, and to find out the likelihood estimation. So likelihood estimation says that we first construct a function by multiplying the PDFs of different variables. If they are IIDs, then it will be just a product of these PDFs, and after finding out the likelihood function, when you differentiate this function with respect to the parameter. So let us suppose we first find out the likelihood function, then differentiate this function with respect to parameter, then you can find out the estimate of the parameter. So this is the first approach. And then second is called method of movements. So method of movement says to estimate the parameter, what we usually do, we equate the population parameter with the sample values. For example, let us suppose we want to find out the value of the mu, and how we will find it out. The easiest way that we usually consider is that we equate this mu with the sample value, that is called mean. So whatever this mean value is coming from the sample, we equate this value with the population parameter, and we can say that this will be our population parameter value. Okay, so there are two different approaches. The first approach says that to estimate the parameter under this frequentistic method, we first find out, if we know the distribution of the data or the variable, then what we usually do is we take we multiply the PDF n number of times if we have n observations with us, and to estimate the parameter, we differentiate this likelihood function with respect to mu if we have the mu parameter with us, and then equate is equal to 0. This equation will give you the estimate of the parameter. Okay, whatever the value we got, that will be come up by solving this expression. But the second method says to find out the information about the data set, about the mean or characteristics of the data set. What we usually do is we equate the population movements with the sample movements. Okay, the next thing is called here the example is given to how one can find out the parameters by first, second, third and other orders of movements. So that is easily we observe that if we have two parameters mu and sigma square, so I already told you that you have, because mu represents the mean, so we are equating the population parameter mean with the sample formula for the mean. That means taking all the observation dividing by the number of taking summation of all the observation dividing by the number of observations, and to find out the second order movement. So this is called the second order movement of sample. So in which we take the square of each observation and take the summation of all the observation divided by the n. That will give the uh, mu square plus sigma square. To find out the value of sigma square, we have to subtract the value of mu square from this expression. That will give the value of variability, right? So here now it is very another thing that is required for discussion, that is called confidence interval. So in the starting I already discussed with you that in this inferential statistics we have two things. One, we have further two segment that is called estimation, testing, or hypothesis. In the estimation, we have further two branches. One is called point estimation and second is called confidence interval. Okay, so the difference between these two techniques is that in the point estimation, when we have a sample with us and we want to estimate the parameter based upon this sample, right? So it means based upon few observation we want to estimate the parameter, and we got a single value. Okay, and in this confidence interval our main objective is that why it is required as compared to this point estimation. The reason is that if we keep on changing the sample, so let us suppose I have few observations from this sample. So this sample is coming from this place. So based upon that, I got the value of mu. And if I take, if I you or you can say take another sample from this place, I got the another value of mu. So there is a variation in the value of mu. So after that, so to you can say to get such value of mu that lies in this interval, so we go for confidence interval. So it means, so we need such value of mu, or you can say we need a lower and upper value of upper limit of mu, so that you can say that your value of mu lie in this interval. So for that we use confidence interval. Okay, so by using the concept of confidence interval, we are able to remove the drawback of this point estimation. Okay, so coming back to that one. So now you can see that, so whenever we are interested to find out the parameter, so we generally consider example that if we have this parameter mu that is for normal distribution, so we have this expression. Okay, so that this expression says that, so we have a test statistics, that means a mathematical form that is used to find out this range of mu. So this range shows that, so your true parameter, population parameter mu lie within this interval. So whether you change, whether you take any sample, whether you choose any you can say observation, your true parameter mu lie within this interval. So and that is very important. Okay, the reason is because while conducting any clinical trial, our main objective is that whether our result is relevant for a particular group of people or whether it is applicable for all the persons. So if we observe a value of mu by using this confidence interval, we will get a clear understanding that the range of the mu lie within this interval. So you can use that for further analysis. Okay, so the next approach is that is called Bayesian approach. So now I'm coming to the actual thing. So these the things that I have discussed earlier, that is required for the discussion of this technique, that is called Bayesian approach. So now I'm going to discuss with you what is prior distribution and how this prior distribution, I'm going to relate this prior distribution with the concept of Bayes theorem. Then what is posterior distribution? Then we will discuss about the loss function, then expectation of posterior. So first come to the concept of prior distribution. We let us suppose we have a variable x in a random sample found population specified by the density function. Okay, and we have a parameter theta, right? So as a result of the parameter theta being a random variable, so now in spite of considering theta as a constant, so we have a theta as a random variable. So when we assume anything that is a random variable, so that must have some either the mathematical form of that function is known to us. So it must be it is assumed that it has some distribution. Okay, This allows the use of knowledge, any knowledge available about possible values for theta before the collection of any data. So here, what we're doing, in spite of considering that our x follow any distribution with the parameter, so here we are not considering these parameters as a constant. So we are assuming that these parameters again a random variable, and we our main concept is to find out the distribution. So if we are from the values of the theta, if we are able to identify this can be the distribution of theta and other things, then it is good. Otherwise, we generally assume a discrete uniform distribution. Okay, or this allows the use of any knowledge available about possible values of theta before the collection of any data. This knowledge is quantified by expressing it as the prior distribution of theta. Okay, so the prior distribution summarize when we know about theta, but before we collect any data set from the relevant population. That is very important. So then after collecting appropriate data, the posterior distribution is determined. So it means that, so we must know first the prior distribution of theta. Okay, and if we have the knowledge of the prior distribution of theta, then we are going to collect the data set, and after that what our main objective is to find out the posterior. So what is prior and posterior? Prior means that information about the distribution of theta that is known to us in advance. Okay, and posterior means after collecting the data, so how our data set impact the value of theta, so that is called posterior. Okay, so the Bayesian theorem, the Bayes approach combines the sample data with the prior distribution. The conditional distribution of theta given the observed data is called the posterior distribution. So it means, so if I assume that my theta is a random variable, so it follow a distribution that is called, so the distribution of theta is called prior distribution, and I have the knowledge of that x given theta. So because I already assumed that theta is a random variable, so for defining the distribution of x, so I have to specify a specific value of theta. That means I have to assume that for a specific value of theta, I can say that it again follow another distribution. So if I assume that it follow normal distribution, okay, with parameter theta and theta square. So this is just a you can say for representation purpose. So here our main objective is now to find out the distribution of theta given x. So now you can recall from the concept of Bayes theorem that in the Bayes theorem we have the information given to us priorly. So if you remember from the example of manufacturer, right? So we have three manufacturers, 1, 2 and 3, right? And it is given to us that how what is the percentage of garments that each manufacturer produce. One produce 60% and other produce I think 20% or 30%, and then the remaining one is 10%, right? So here you can see, so it is already given to us that this manufacturer produce 5% defective items, this produce 50%, this produce 10% and 50%. So now what we are doing, we have prior information given to us and we have data set with us, and now what our main objective was to find out the probability that we got a defective item, we got a faulty garments, and what is the probability that it is coming from the third manufacturer. So what we are doing, first the information that is given to us that the probability of manufacture that it produce a faulty item is given to us, but our main objective is to find out the probability of faulty, that we receive a faulty item and it is coming from the first manufacturer or third manufacturer. So what we are doing, we are finding out the opposite. That means what are the events that we have here, we are finding out the opposite case here, right? So here in this Bayesian statistics, in spite of considering the events, we have the parameters. Okay, so generally we assumed our parameters as a constant. So if this is not the case, if our parameters again a random variable, then how we will proceed further? That is important thing. Okay, so we already have discussed that here theta that represent the parameter, that is not a constant here, that is again a random variable that follow another again a distribution. So after that we have another case in which after defining the distribution of this parameter, we have this case that we observe the values of data by considering that they follow distribution by assuming a fixed value of theta. So now our main objective is to find out the distribution of theta given x. Okay. So come to the first thing, that is called prior distribution. So prior distribution may be discussed, or you can say previously study, and before a trial is carried out. So I already discussed with you before conducting a trial. So most of the researchers have some idea about that how or you can say what type of patients they have, what type of study they will conduct. They have some information regarding the effectiveness of the treatment, right? Such as the drug response and toxicity probability or certain dose level, or the patient's median survival time in certain groups. So they have some prior information in terms of these cases like toxicity probabilities or survival times in certain groups. So these are the prior information that they have. So it can be also be written in understand in this way. What are the typical responses and toxicities for this patient population under treatment? So these questions will help you to understand what is prior information, what percentage of patients will experience each of the clinical meaningful event, what is the highest dose that has negligible toxicity. So these are the prior information that they have. So how this information will be incorporated in our study, we will discuss that. So in our study, it can be further categorized into four parts. One is called non-informative prior. So what is non-informative prior? Already discussed with you that when there is no information given to us in advance. Okay, so we generally assume that this can be considered as a prior distribution in this case. So non-informative prior refers to a flat or vague prior distribution under which the posterior distribution is approximately proportional to the likelihood. What they are trying to say, I have just discussed with you that our main objective is to find out the distribution of theta given x. Okay, so what information we have? We have the information of x given theta, and we have the distribution of this theta, right? And if you want to summarize this function in this way, that this is the case. Okay, so here what they are trying to say. So refers to the flat or vague prior distribution under which the posterior distribution is approximated. So this is called posterior distribution. Proportional to the likelihood. So likelihood is defined as a product of values of x. So if I write down this function of likelihood here, and so the data fully dominate the posterior distribution. So what they are trying to say. So if we do not have any prior information about the data set, so here if we assume any distribution, so after multiplying any constant terms, we will get a new form of the distribution. So that will impact the whole study, or you can say that will dominate the posterior distribution. Okay, so the second case is called skeptical prior. So first let me help you to understand the concept from this graphical representation. Now you can see that when there is no prior information is given to us, that is called non-informative, in which we assume that each and everything has equal weight, right? So we are assigning equal weight to each and every event, okay, that we have in our study. Okay, in the skeptical prior, we have some tendency that we, there are some rules or regulation, or you can say legal foundation that we have. So we incorporate such distribution that satisfies such type of you can say, or they have the PDF of such type of form that is used for this case. And when we talk about optimistic, so it means, so it is called when we have a positive skewness cases, that comes under skeptical. And when we have negative skewness, that is comes under optimistic. And when we have a you can say bell shaker, that comes under clinical. So here uh, let me discuss with you this concept through this you can say interpretation. So skeptical priors is more conservative prior, which often represent the standing point of the regulatory agency. So it means when our regulatory agency have some rules and regulations, we have to satisfy all of them, then we consider such type of prior distribution. So when we have optimistic prior is more enthusiastic prior distribution, which is often dominated by the investigator optimism. So it means, so when the investigator is very optimistic about a particular opinion that this drug is very effective in that case, okay, this put most of the prior probability mass at favorable values of the treatment effect. And then we have clinical prior is a relatively objective prior distribution which tries to eliminate the potential subjectivity by taking an average of the several prior distributions. So we already know that clinical trial distribution seems like a normal bell-shaped curve, and it is easily observed that the shape of the curve can be modeled by the normal distribution. So the reason behind is because it is observed as an average of several prior distribution, and we already know that central limit theorem says that most of distribution approaches to normal distribution for large sample size. So this is the case. So I already told you that how to find out the posterior distribution. So for that, we are interested to find out distribution of theta given the data value, and for that we first have the values of x by assuming a particular value of theta, and then we already know the prior distribution of theta. By using this mathematical equation you can find out the distribution. So how why we need likelihood function, that for that I will discuss again that this form that is called f of x given theta is basically a likelihood function. Okay, so we have to multiply the likelihood function with this f of theta and divided by the likelihood function this f of x, then only you can find it out. So here one can also you can say observe that sometimes it is not always the case that you can get a complete form, or you can say is the final form of this posterior distribution in the terms of a particular distribution. So there are few examples here that I will discuss, that in which we can see that the final expression of this posterior distribution does not belong to any distribution. So we generally assume that proportionally this expression follows some particular distribution. Okay, so already told you that we have a prior distribution, we have a likelihood function, and C represents a constant term. So that this distribution will be satisfy the condition of this PDF, that when we integrate our posterior distribution over this range, then the probability is coming as 1. Okay, for example, we have a example here. So annual number of patients visit the hospital follow a Poisson distribution with mean mu, and the prior distribution of mu is exponential. So here what I am saying that, so we have a exponential distribution whose mathematical form is this one, e minus lambda lambda x upon x factorial. So here in place of lambda we are assuming mu parameter. So here it is e minus mu x upon x factorial. So here we are observing this value of mu not a constant, we are observing value of mu as a variable. So mu follow again in distribution that is called exponential. Okay, so the information is given to us that in the previous 2 years the number of patients visiting were of 26 and 28 respectively. So this so we have the number of patients with us. So you can see that the prior distribution of mu is exponential, and the mathematical form is given here. And to find out the likelihood function, we have this expression by putting the value of this x in the PDF of this Poisson distribution. So we got this form, right? So here you can see that we have some terms in terms of factorial here. Okay, so we are considering all these constant terms in this phi. Okay, so we use this c that represents the constants of, so you can see, so c into e minus 2 mu and mu power 54. Okay, so here if someone is familiar with this the distribution, the mathematical form of the distribution I already discussed with you. So if someone remember that while discussing the distribution of gamma, okay, so we have this form, right? Okay, so here mu is a variable, so you can, if you can match these two you can say the PDF, the mathematical form of distribution, so you can say that after doing some simple you can say modification in this mathematical form, one can say that the posterior distribution of this mu is coming from the gamma distribution. So now you can see that our initial distribution of variable x is Poisson with some parameter mu, and mu follow a distribution that is called exponential. So when we are trying to find out the distribution of mu given x, so that is coming different, that means that is coming a distribution that is different from these two distribution. So that's why it is called posterior distribution. So why we are doing why this theory is developed? The reason behind is that because here we are observing that mu is a variable who has certain characteristics, but when we incorporate the information from the data, okay, and trying to find out the distribution of mu, so that follow another different distribution from this exponential distribution. So it means now it is able, or this distribution is more capable to you can say model the situation very well as compared to earlier. So this is the whole idea behind Bayesian statistics. And in this clinical trials, so we are using this concept. The reason behind is that because in the clinical trials we have some information regarding the patient, we have some information regarding the drug, we have some knowledge about the you can say side effect of the drug. So if we incorporate that information while estimating the parameters, so we will get a new form of the distribution that incorporates more information that is more relevant as compared to the earlier case. Okay. So there is another concept that is called conjugate prior. What is conjugate prior? So in the previous example, I have assumed that mu follow exponential distribution, and our x follow mu that follow Poisson distribution. Okay. So if in spite of this case, if let us suppose I have such distribution G, and x given mu that follow another distribution that is S. Okay, if I am trying to find out the distribution of mu given x, if it is again coming the distribution G, but with different parameters. So let us suppose in this case of distribution G, I have the parameter alpha 1, alpha 2. Okay, but when we incorporate the information of x and find out the distribution of mu given x, I got another form of this parameter alpha 1 dash and alpha 2 dash. But so it means the distribution is same, but the parameter value will be different. So those distributions are those priors are called conjugate prior. So it means whatever the distribution that you have assumed in the prior, that there will be no change in the distribution, but there will be only change in the parameters. Okay. So we have one example. So suppose that x1, x2, xn is a random sample from the type 1 geometric distribution with parameter p. So if all of you are aware about the geometric distribution, the geometric distribution is that in which we repeat the experiment till the time we will get the first success, right? So it means if we conducted x experiment and after that we will get the first success, so it means the distribution is defined as in this way, or it is can be written as p1 minus px minus 1 into p, right? So here p value lies between 0 to 1. So we will show an example that this geometric distribution is an example of conjugate prior, in which if we consider the distribution of x, that is any other distribution. Okay, so let me help you to understand this example. So okay, so here it is given that x1, x2, xn is a random sample from the geometric distribution with parameter p, and here define a family of distribution for p that would result in a conjugate prior. So we have to find out the distribution of p that can be considered as a conjugate prior. So x follow geometric distribution, but our main interest is to find out that distribution that can be considered as a conjugate prior. So it means whatever the distribution our p follow, so after you can say incorporating the information that we get from this data set, so p given x follows same distribution. Okay, so here we have this example with us in which we first have the PDF of this geometric distribution. After that we have to find out the likelihood function. Then you can see that if when we have to find out the PDF of the posterior distribution, so for that we need this likelihood function, we need this distribution of p, then divided by this likely function of x. Okay, so in spite of that, so what we're doing here, we are multiplying this likelihood function with this prior distribution of p. So it means if anything, if the final result should be the same, that means if we are aware about the distribution of beta distribution, so beta distribution of this form. Okay, so where x lies between 0 to 1. So now you can easily see that. So if we consider the distribution of p as a beta distribution, so after multiplying this likelihood function with this distribution of p by assuming that it again follow a beta distribution, the final distribution will again comes as a beta distribution. So this is, so in this case beta distribution is a conjugate prior, in which you can see that. So if we assume the distribution of this parameter p as a beta distribution, after multiplying the PDF of this beta distribution with this likelihood function, you will get again the form in the final form as a beta distribution. Okay, so this is the whole concept of this Bayesian statistics. I think I should stop here because we have only 2 hours to complete. I will try my best to cover each and everything that is related to Bayesian statistics. So if someone has any question. Hello.
Thank you, sir, for finding time to talk about such an interesting topic. And you beautifully covered that all the topics like fundamentals of clinical trials, primary objectives with example, parallel and crossover design, data collection in clinical trial, and many more. So I think it was very interesting topic, and I I hope and I'm sure that all the participants must be agree with me. And you beautifully explained with making it simple and giving examples. I appreciate and thank you so much. And I would like to request participants to ask questions if any. Either you can send your questions in chat box.
Dear participant, you are permitted to unmute yourself. If you have any question, you can unmute and put your question directly from the speaker.
You can also send me questions on my email ID. So kindly share my email ID to them. And if someone has any query regarding this topic, I will be happy to answer that questions.
Thank you, sir. So participant can ask questions through email to Dr. Harmanpreet, and if there is any question for now, you are most welcome to ask. Okay, I would request Dr. Meherchand to kindly share certificate as a token of appreciation for the contribution of Dr. Harmanpreet Singh in the workshop.
So thank you, Dr. Meherchand. Actually, this topic is very vast, and 2 hours is very less to explain this concept.
So even then, you probably, I think all will agree that you have tried to explain in a very simple way with giving examples, and it made it interesting too. So thank you so much for your time.
Thank you.
And I would like to thank all the participants for being the part of the workshop. Thank you so much. Thank you, everyone. See you tomorrow at the same time. Okay. Thank you.
Thank you.